Hello, students. Uh, Good afternoon. Can you hear me? So the only problem is the polls, right? So, anyways, please respond to me in the chat box as usual. If my voice is clear and the audio is well for you, as soon as possible, that would be very helpful. Just a quick checkup on audio and video. Please let me know if my audio and video is clear for you. And people in the YouTube, you can use the chat box to communicate. Okay. You can just let me know if you have any problems with technical issues. Okay. Okay, I will use the chat box. Sorry, can you hear me well? Students, can you hear? Can you hear my voice clearly? Otherwise, it's hard to start soon. Please let me know. Okay, I'll do one thing. As usual, I'll start the session because there might be some technical glitches as we go to the end of the session. Okay. So I will start my session right now. So let's see. If you have any problems, you can just put it in the chat box so we can correct it as soon as possible. Okay. So do you remember in last class what we discussed? We discussed about glycolysis, right? We had an elaborate session in discussing the process of glycolysis, right? So we were breaking down the process of glycolysis, and by the end of the glycolysis process, we got one of the resultant, that is pyruvate, right? Or pyruvic acid. So now we can use the pyruvate, like living organisms use the pyruvate in different means based on the availability or based on the requirement of oxygen. So whenever we talk about the dependability on the oxygen, we can divide our respiration process into two different means, right? That is, one is anaerobic respiration, other one is aerobic respiration. See, glycolysis is not the end of utilization of glucose. There is even more, elaborate pathways that are going to come up next. Okay. So now what has happened here? Till now, we have produced one of the byproducts of the glycolysis, that is pyruvic acid or pyruvate. So using this pyruvate, as I said, whether there is oxygen requirement or organisms which do not need oxygen. And the second point is organisms which strictly need oxygen for the utilization of this pyruvate to produce more amount of ATP molecules. Understood? So let us see the first case, that is anaerobic reactions. Okay. So in anaerobic reactions, what is happening in anaerobic reactions? Okay. Oh, sorry. Before we head on to this topic, you have the first question. Okay. Very simple question. Incomplete oxidation of glucose into pyruvic acid with several intermediate steps is known as TCA pathway, glycolysis, HMS pathway, and Krebs cycle. So you can actually type your answer in the chat box. I'll be waiting for your answer, and I will give you exactly 1 minute only. Okay, exactly 1 minute, very strictly. So your time starts now. People, it's from 1988. It's very simple question. Come on, no. Incomplete oxidation of glucose into pyruvic acid is with several intermediate steps is known as what? Okay, we have an answer now already. Arey yaar, I need answer, not good afternoon. Okay, fine. Good afternoon. Please let me know. Faster students, like I will give you 15 seconds now. Just put an option. If you don't want to type the entire thing, just put an option in your chat box. And YouTube students as well. So we have 5 seconds. 5, 4, 3, 2, 1. Yeah, B. So we have the first answer. Thank you. So one of your friends have answered it is B, that is glycolysis. So all of you agree with this answer that B is the correct answer for this question. Actually, when we talked about oxidation of our glucose, basically it started with the unavailability of oxygen, right? So enzymatic oxidation took place, but is it the complete breakdown of glucose happened? Definitely no. It only stopped at pyruvic acid. So the resultant pyruvic acid we get from which of the process? Glycolysis, right? So our answer is B, glycolysis. It is not TCA pathway. It is not HMS pathway. And Krebs cycle we are going to study in further, okay, when it comes to aerobic respiration. So our answer is B. Understood? Okay, fine. Okay, let's get back to our topic now. So in our topic, as I said, when we have a situation where there is no oxygen, right? So when there is no oxygen, it undergoes anaerobic respiration. So our pyruvic acid can be utilized in two different ways. Okay, that process is called as fermentation. So in fermentation, we have two different types of fermentation. One is called as lactic acid fermentation, and the second process is called as alcoholic fermentation. Correct? No, when there is no availability of oxygen, we can expect there is two different pathways that can happen. One is lactic acid fermentation, other one is alcohol fermentation. Now let us talk about your lactic acid fermentation first, because it is very easy to understand for us. Okay. When it comes to lactic acid fermentation, imagine that you are running on a treadmill or you are having a very tedious day on your ground. Your PE teacher is making you run a lot. So now what happens as you keep on running? Now you're gasping for more and more air, isn't it? So when you're gasping for more and more air, why is your breathing increasing more and more? Because your tissues are using lot of energy, lot of oxygen for breakdown to provide your tissues to construct, your muscles to construct. You need more energy, right? More energy means you need more oxygen. More oxygen means you need to breathe more. And moreover, there is more carbon dioxide in your tissues that has to be put out. So during this process, you can't breathe like very high levels. Like you can't keep on increasing your rate of respiration. No, there is a limit. So when that limit crosses and your body is not getting sufficient oxygen, what is happening to all the pyruvic acid that is being formed in your tissues? It is being converted into lactic acid, correct? No? When the lactic acid accumulates in your tissues, you get a tremendous amount of pain in the form of cramps. Muscle cramps will happen, correct? No? So this lactic acid accumulation or lactic acid fermentation mostly happens with respect to aerobic organisms. Only very, very few organisms are known to undergo when it comes to microorganisms like yeast, okay? Only very, very few organisms undergo this type of fermentation when it comes to microorganism. Which one? Lactic acid fermentation. And the second type of fermentation is alcoholic fermentation. So we have a very wide range of this particular fermentation, correct? So alcoholic fermentation is simple. So whenever there is absence of oxygen, pyruvic acid is converted into alcohol, okay, or ethanol. So this is called as alcoholic fermentation. Understood? So in either case, it is very, very dangerous. Okay, the fermentation process is very dangerous for a living cell. So let us see what is happening here. Once your glucose is broken down into pyruvic acid by the process of glycolysis, correct? No. Now here, as I said, your pyruvic acid is converted into either ethanol and carbon dioxide or lactic acid. Understood? So now for this process, who is helping this process to get accomplished? There is an enzyme called as pyruvic acid decarboxylase, okay, and other one is called as alcohol dehydrogenase, okay, or we can also call it as lactic acid decarboxylase is another enzyme. Okay, lactic acid decarboxylase is one enzyme, and another enzyme is called as alcohol dehydrogenase. Understood? So now what is happening when these two enzymes act? A catalytic reaction will take place. Now what is happening when there is pyruvic acid, and then we have an enzyme called as lactic acid dehydrogenase, and lactic acid dehydrogenase, okay, will help reduce NADH, okay, will help in will help in reduction of NADH to NAD plus. During this process, pyruvic acid is converted to lactic acid. Understood? So that is how the lactic acid accumulation will take place. Understood? And next one. Here, the same pyruvic acid in the presence of alcohol dehydrogenase, and the reduction reaction will take place because of this enzyme alcohol dehydrogenase. It will reduce your NADH plus H plus into NAD plus. So that will result in the formation of ethanol, okay, carbon dioxide. Understood? So now undergoing fermentation reaction will result in what? So when the organism undergoes fermentation, so there will be production of ATP, and the amount of ATP produced for one glucose molecule through fermentation is 2. Okay, only 2 ATP molecules will be produced. Overall ATP molecules from glycolysis it is 2, correct? No, and from fermentation it is 2. So 2 plus 2 is 4, correct? No, so the amount of ATP utilized in glycolysis it is 2, isn't it? So now you have, by your by your fermentation process, you will get only 2 ATP molecules. Understood? Now fermentation is really bad. Why it is bad? Because whenever the cells are accumulating more and more ethanol, that can lead to the death of the cell. Why? Because alcohol is a very high dehydrating agent. It will remove very high amounts of water from the cells. Okay. Now, when the alcohol percentage is increasing in the surrounding, the cells will lose a tremendous amount of water that can lead to cell death. That is why whenever you talk about yeast, take an example of yeast. Yeast, when it keeps on fermenting, when we use yeast for the preparation of wine, right? So when the yeast keep on fermenting, and if the alcohol percentages reach above 13%, the cells will die. Okay. But when we take about different alcoholic beverages that we have, some of the alcohol will be of 38%, some of the alcohol will be of 15%. Okay, but they're reaching above 13%. Now go and research how, the small, curiosity, what is the process that we undertake in fermentation of alcohol, and how high amount of alcohol percentage is achieved by fermentation through the same cells, which cannot reach 13% and above. Okay, so that you can understand by. So in conclusion, fermentation can be concluded as the glucose is converted to pyruvic acid by glycolysis. Okay, then pyruvic acid in the presence of alcohol decarboxylase, lactic acid decarboxylase, and then you have another enzyme called as alcohol dehydrogenase, where NAD is reduced to NADH2. Here what do we get? We get ethanol plus carbon dioxide, or we'll get lactic acid plus carbon dioxide. Understood? So this is called as fermentation process. Understood, right? So now we understood the alcohol production. So the same thing is happening even in lactic acid. I told you, in mostly animals which use oxygen for the for the aerobic respiration, whenever the respiration quotient is not sufficient to provide your enough amount of oxygen for the breakdown of your glucose or pyruvic acid, then your muscles will switch into anaerobic respiration. So during anaerobic respiration, lactate dehydrogenase is an enzyme which will convert your pyruvic acid into lactic acid. Okay, and the reducing agent for that is NADH plus H plus. It is reoxidized to NAD plus. Okay, in both the types of fermentation, correct? No. So in both alcohol fermentation as well as lactic acid fermentation, so this is happening. So now what happens? The lactic acid is again pumped back to your liver. Okay, so your lactic acid cannot be utilized by your muscle tissue. It will be pumped back to your liver, and the liver will convert into pyruvic acid, and again the pyruvic acid undergoes aerobic respiration, and then the utilization of energy will be completely done. So that is why we get muscle cramps. Understood? So this is about fermentation, correct? No. So what are the drawbacks of fermentation? First one we already understood. When it comes to lactic acid or alcohol, now because of the toxicity, it can lead to the cell death. Understood? And another important aspect is, if you see the amount of energy output of this fermentation reaction, the energy produced is very, very limited. Okay, how much energy is produced? Only 7% of the energy output will happen for every glucose molecule that is utilized. Understood? So only 7% of the energy will be trapped in the form of ATP, and 93% of the energy that had to be produced will not be produced. Okay. So that is the drawbacks of fermentation. Two drawbacks. One is the energy output is very, very less. And the second one is the products, that are byproducts of your fermentation, can be very hazardous and can lead to cell death. Understood? So this is about fermentation. Fine. So did you understand what is fermentation reaction? Two important aspects. NADPH is reoxidized into NADH, and the two important enzymes is alcohol, okay, dehydrogenase, lactate decarboxylase are the two enzymes that are used, and the net products are only very little amount of ATP, that is only 2 ATP is produced. Understood? So this is about your anaerobic respiration. If you have any doubts, you can put it in the chat box. Okay. So let us go and talk about aerobic respiration. So before we start the topic about aerobic respiration, so you have your first question. Aerobic respiration respiratory pathway is appropriately termed as parabolic, amphibolic, anabolic, and catabolic. So here you have your option. And the answer we already got. One person is telling parabolic. Okay. Come on, people, you have 1 minute. So we have enough participants. You can answer the question. So the students from the YouTube, you can try responding on your chat box. Okay, you can use your chat box to respond. I will watch your answers too. Okay, so interact so that we will understand you are following the class properly. Others, what is your answer? Make it quick. Another 20 seconds. 2009 question, neat question. According to you, when it comes to aerobic respiration, so what can it be termed as? What is it termed as? Is it an anabolic pathway, catabolic pathway, or parabolic pathway, or amphibolic pathway? Okay. Take a guess at least. Okay. Can we continue? So aerobic respiration pathway is appropriately termed as what they have asked. No, it is called as amphibolic pathway. Amphibolic pathway means it includes both catabolic and anabolic pathways combined. It is not just construction or destruction. Both construction and destructive processes are happening in aerobic respiration. That is why aerobic respiration is called as amphibolic pathway. Understood? So for this question, the appropriate answer will come in while we discuss this particular chapter. Okay, as we move on. So you have one more question now. EMP, you know what is EMP, right? EMP is also called as what? Glycolysis? No. EMP can produce, okay, hold on. Okay. EMP can produce a total number of 6 ATPs, 8 ATPs, 24 ATPs, and 38 ATPs. What is your answer, people? Tell me. EMP pathway can produce a total number of how many ATPs? EMP is also called as glycolysis, no? So it will produce how many ATPs? Do you remember the process of glycolysis? Yet you have doubts? Okay. So, fine. No problem. We'll solve this question, okay? So EMP pathways is glycolysis, no? In glycolysis, what happens? We will invest 2 ATP molecules and finally we will get around 4 ATP molecules directly. But another 4 ATP molecules is we get by reduction of NADH, okay? So when we do the reduction of NADPH, so we will get another 4 more ATP molecules. So the total amount of ATPs that are produced by the EMP pathway is around 8 ATP molecules, okay? It's not 24, it's not 38, it is not 6. The total number is 8 ATP molecules. Understood? So you have to remember this number of ATPs. It's very important. Even though you don't remember the enzyme names, it's okay to a certain extent. But most questions are based on how much, what is the net result of this particular process, no? So how do we get this 8 ATP? Whatever the NADH that is being produced, no, NAD is like reduced to NADPH. So after the reduction of NADPH, now again this NADPH will be utilized in the Kevin cycle. So in this Kevin cycle, the same NADH will give rise to your 2 ATPs, okay? So 2 molecules of NADH will be produced. Now, if you go back to your aerobic reaction, come back here, okay? Oh, it is not there. Sad. Okay, oh, here it is. See here, you can see that there is NADH that is being produced. No, the same NADH will be utilized in the Kevin cycle. Also, okay. So here what happens, this NADH will be used for the production of ATP molecules. So the number of NADH that will be produced is, 2, correct, right? So by the end of the process, they will give 4 ATP molecules. 4 plus 4 is 8, okay? So you have the answer. The answer is 8 ATP molecules, okay? Here it is, very, very clear, okay? See, the total number of NADH is 2. So this will give rise to how much? See here, the net product is 2 ATP from the glycolysis, correct? No. So actually what is happening? Here we use 1 ATP molecule. Here we use 1 ATP molecule, correct? So here we get 2 ATP molecule. Here we get 2 ATP molecule. 2 plus 2 is 4. 4 minus 2 is 2. We have 2 here, correct? No. Now we have 2 NADH molecule here, correct? No. So these 2 NADH molecule will give us how many? Will give us 2 and 2, 4, 6 happened. And then again from 2 pyruvate, correct? No. So we will have around 8 ATP molecules by the end of glycolysis. Understood? Okay. So that is your answer. So let's go further and discuss about more topics. Fine. Now, what is aerobic reaction? Understood? So what is happening in aerobic reaction? In aerobic reaction, there is complete oxidation. Okay, complete oxidation of your pyruvate in the presence of oxygen that releases carbon dioxide, water, and energy. So this process is happening in mitochondria. Understood? So now what is happening? Once we have the pyruvate as a final product of the glycolysis, it is transported from cytoplasm into the mitochondria. Okay, that is the first process. Now what is happening? We have glycolysis. We got 2 ATP from here. And the byproducts of this glycolysis is also very important. NADP, right? NADPH. So this is also transported into your mitochondria. Here, what is happening? The pyruvic acid will undergo this elaborate pathway called as Krebs cycle. Okay. And then what is happening? There is a reduction of your pyruvic acid. And during the reduction of your pyruvic acid, now what happens? There is release of hydrogen. So this hydrogen is used as an electron and it is moved across in the form of electron transport system. and the resultant of electron transport system will be used for synthesis of high amount of ATP molecules. Understood? And also hydrogen that is used for the movement of electrons will be removed in the form of water molecules. Understood? So here we will not talk about your electron transport system now. First, let us complete till your Krebs cycle. Okay, let us complete till our Krebs cycle. Okay, so let us see what is happening in our aerobic reaction. Okay, so first what happened through EMP pathway, correct? No, glycolysis happens in EMP pathway in cytoplasm, correct? Now what is happening? Once it is complete in the cytoplasm, it is transported to your mitochondria. In mitochondria, what is happening? Oxidative decarboxylation happens in the mitochondrial matrix. If you see your mitochondria structure, okay, this is the outer membrane, this is the inner membrane. Inner membrane is folded into different foldings, and this is called as cresta, correct? No. So this cresta, within the cresta, you have your mitochondrial matrix, where we call it as oxidative phosphorylation reaction is happening, where the ATP production is happening, correct? So first what is happening when your pyruvic acid is entering into your mitochondria, here oxidative decarboxylation will happen. Understood? What is oxidative decarboxylation? Slowly the number of hydrogen atoms that are present in your pyruvate will be removed one by one. Understood? So as it is removed one by one, it will be converted into subsequent byproducts. That is what is happening. Understood? So that is happening in mitochondrial matrix. What is the third step? Whatever we have the oxidative decarboxylation of pyruvate that happens, that will enter into the Krebs cycle in the form of oxaloacetic acid. Okay, or you will understand about it. Okay, it will undergo Krebs cycle or TCS cycle. Understood? So after undergoing TCS cycle, this cycle is also happening in where? Mitochondrial matrix. So the resultant of this TCS cycle, okay, further will undergo oxidative phosphorylation, where your ATP molecules will be produced through electron transport chain. Understood? So first step, which is the first step of our reaction that is happening in our mitochondria, that is oxidative decarboxylation. What is oxidative decarboxylation? Your pyruvic acid, correct? No, that is extracted from your cytoplasm. After the glycolysis, no, it is converted into acetyl-CoA. Understood? So for this process, there are several coenzymes and NADP plus requirement is there. Understood? So that is why we write pyruvic acid in the presence of coenzyme A and NAD plus as the reducing agent, okay, as an oxidizing agent, sorry. Now what happens? In the presence of magnesium, very important, you have to remember this, in the presence of magnesium and the enzyme pyruvate dehydrogenase will convert our pyruvic acid into acetyl-CoA, okay, and the resultant of conversion would be, you see here, it will result into carbon dioxide, and NAD plus will be oxidized into NADH plus H plus. Understood? This is a very important cycle. Now once the pyruvic acid is converted into acetyl-CoA, this acetyl-CoA is used for as the first product for the TCA cycle, or it is also called as Krebs cycle. Did you understand, students? I will just make a quick summary. You can understand. Okay. First, what did we have? Glycolysis. Out of glycolysis, what pathway it is? EMP pathway, correct? Where did it happen? Cytoplasm. What is the resultant of this glycolysis? Pyruvic acid. Pyruvic acid is going under what? Oxidative decarboxylation. Once oxidative decarboxylation is happening, where it is happening? Mitochondrial matrix. After oxidative decarboxylation, what do we get? We get acetyl-CoA. Where does this acetyl-CoA is used? Acetyl-CoA is used for Krebs cycle or TCA cycle. Understood? So where is this TCA cycle happening? Matrix of mitochondria. After we have this, then oxidative phosphorylation will happen for the production of ATP. Understood? So when we are talking about oxidative decarboxylation of pyruvic acid, I tell you, it involves several enzymes, NAD plus, as well as a pyruvate dehydrogenase for this process. And this process happens in mitochondrial matrix. this region. Understood? This region of mitochondria it is happening. So how does it happen? Your pyruvic acid will react with the coenzyme A plus with with an oxidative agent like NAD plus in the presence of pyruvate dehydrogenase. It will result in the formation of acetyl-CoA, carbon dioxide, NADH plus H plus. Okay. Now this acetyl-CoA which is extracted from the pyruvic acid will be used for TCA cycle. Understood? Okay. So now you can see very clearly what is happening, correct? No, in the process of your oxidative decarboxylation of pyruvate, correct? Right? So pyruvate is complex and it is a large subunit. So now on this in the mitochondria, we'll have to undergo series of steps. You can see here this particular structure, correct? No, a carbon molecule is removed. That is why we call it as decarboxylation. When a carbon molecule is removed, a carbon dioxide comes out, correct? No, a CO2 molecule is removed out. For this process of conversion of pyruvate into acetyl-CoA happens like this. Understood? Yeah. So the details of this particular enzymes is not necessary for now. All you have to remember is what is oxidative decarboxylation of pyruvate. A 3-carbon atom of pyruvate is converted into 2-carbon atom of acetyl-CoA, as simple as that. And during the process, a carbon dioxide is removed, okay, and the acetyl-CoA is produced, and then NAD plus is oxidized to NADH plus H plus, as simple as that. Understood, people? Okay. So let us go for what is citric acid pathway, or it is also called as Krebs cycle, because Krebs is the one who discovered this pathway. It is also called as TCA cycle. Why? Because tricarboxylic acid cycle. Okay, that is a name for this. Understood? So let us see elaborately what this pathway is and what is the resultant of this pathway. Understood? So now, before we start the pathway, okay, now your acetyl-CoA that is being produced, correct? No, acetyl-CoA that is being produced, actually this acetyl-CoA is converted into oxaloacetate. Okay, OAA we will call it as. Okay, oxaloacetate is the first member. Understood? So this oxaloacetate, actually what happens, a water molecule is added to convert this oxalic acid into, see, if you come back here, the resultant is acetyl-CoA, isn't it? So this acetyl-CoA that we have to use, correct? No, will be actually converted into oxaloacetate. Understood? So this oxaloacetate will be converted by citrate synthetase, okay, into, see, actually what happens, this oxaloacetate is there, right? Oxaloacetate will be converted into citric acid basically. So that happens by a series of conversions of this citric acid. Okay, see, for example, we have oxaloacetate in the presence of citrate synthetase. Okay, by the addition of water, now what we will have, we will get citrate. Okay, again by dehydrating the citrate in the presence of aconitase enzyme. Okay, forget about the enzyme. We have 3 important steps here. I will talk about it. So we usually talk like this. Oxaloacetate is converted into citrate, and citrate is converted into isocitrate. Okay, isocitrate is converted into alpha ketoglutarate, and alpha ketoglutarate is converted into succinyl-CoA, and succinyl-CoA is undergoing series of steps where it will come back to oxaloacetate. So this is what is a general step. So I will break that down into very elaborate way. Understood? So what happened? Acetyl-CoA had to be converted into oxaloacetate by adding a water molecule. So once we add a water molecule, which can be converted into a citrate. Understood? By the enzyme called as citrate synthetase. Understood? So once we have citrate synthetase, now the citrate synthetase will undergo series of conversions, like isomerisms will happen. Okay, see, citrate is converted into cis aconitate. That means this citrate is actually isomerized, correct? No. So by what? By removing a water molecule. Again, this cis aconitate is converted back into isocitrate. The main aim is we had to convert this citrate into isocitrate, so that will not happen directly. So to convert this citrate into isocitrate, it has to undergo two intermediate steps, as simple as that. Understood? So now we have citrate into isocitrate. Okay. Now what happens? There is a dehydrogenation reaction happening. Okay. In this dehydrogenation reaction, you can observe the structure here. Okay. So you can see here the isocitrate will be converted into a product called as alpha ketoglutarate. Okay. It will be converted into alpha ketoglutarate. In this process, what is happening? A carbon dioxide molecule is removed. Very, very, very important step. Okay. So here a carbon dioxide decarboxylation reaction will take place. So where does the decarboxylation reaction will take place? When isocitrate is converted into alpha ketoglutarate. What is the enzyme that is helping here? Isocitrate dehydrogenase. So for this process, who is the oxidizing agent? NAD plus will help as an oxidizing agent, where NAD plus is converted into NADH plus H plus. So with this process, a carbon dioxide molecule will be removed out of isocitrate. Understood? So once we have alpha ketoglutarate, okay, which is basically made up of only 2 carbon atom. Understood, people? It is made up of how many? 2 carbon atom. So this COO minus whatever is there, this is the acidic group. Okay. So don't worry about that. Understood? So now what happens? This alpha ketoglutarate will be decarboxylated. That means again one more carbon dioxide molecule will be removed with the enzyme called as alpha ketoglutarate dehydrogenase. Okay. See, the TCS cycle is about sequential removal of hydrogen molecules from the structure. So while we are removing the number of hydrogen atoms from the structure, it will release hydrogen atoms. See, you can see that there is release of hydrogen atom here. There is release of hydrogen atom. Subsequently, as we are removing, dehydrogenation means removal of hydrogen atom. As we are subsequently removing the hydrogen atom, we are also removing the carbon atoms as well. Okay. So now 2 important points, we have removed the hydrogen atoms. These place where we remove hydrogen atoms, we call it as dehydrogenase enzyme. Remember this. Understood? One is isocitrate dehydrogenase and the second one is alpha ketoglutarate dehydrogenase. In both positions, the hydrogen atoms from the structure is removed. As a result, a carbon dioxide molecule also comes out. Okay. Now, alpha ketoglutarate will be converted into succinyl-CoA or succinic acid. Understood? So this succinic acid actually will result in production of one GTP molecule. Understood? So the succinyl-CoA will result in the formation of one GTP molecule, that is gynosin triphosphate. Understood? So this gynosin triphosphate simultaneously will be converted into ATP. How? So the GTP will be broken down into GDP. Okay. So this GDP plus phosphate will be resulted. So again, this ADP will use this phosphate. Okay. Like this, and it will convert this GDP into ATP. Understood? It is a spontaneous reaction that is happening. Now the succinyl-CoA, whatever is produced, okay, so succinyl-CoA synthetase will be the enzyme that will convert the succinyl-CoA into succinate. Understood? Now once we have succinate, now what will happen? The succinate will be again dehydrogenated. That means again one more hydrogen atom will be removed from this succinate. Okay, with the help of FAD. Okay, FAD will oxidize this process and convert itself into FADH2 in the presence of succinate dehydrogenase. Understood? So FADH is also necessary for us now. So once we dehydrogenate your succinate, more hydrogen atom is gone. So again, when the more hydrogen atom is gone, in this case, 2 hydrogen atom is gone. So that succinate will result in fumarate. Understood? When we add water molecule to fumarate, it will become malate. So finally, again, malate will be dehydrogenated. Okay, in the presence of malate dehydrogenase. Okay, that will be helped by oxidizing reagent, that is NAD plus will be oxidized into NADH, and that will result in H plus ions. Finally, the malate will be converted into oxaloacetate, which is the starting material of TCA, Krebs cycle. Correct, no people? So what is the overall of this TCA cycle. So we can conclude that in this reaction, when we started from acetyl-CoA, a series of dehydrogenation reactions will take place. Okay, that is the first point. So to starting material of this reaction starts from acetyl-CoA is converted into oxaloacetate, correct? No. So when the oxaloacetate as a starting material, oxaloacetate will be converted into citrate. Okay, the citrate will be converted into isocitrate. Once we have isocitrate, isocitrate will be dehydrogenated into alpha ketoglutarate, where we have one hydrogen atom. To this, what is used? NAD plus is used to oxidize as NADH, first molecule. Correct? Done. So one molecule of NADH plus H plus is produced. Again, once we have alpha ketoglutarate, again it is converted into succinyl-CoA by again one more dehydrogenation reaction. So here also again one more NAD plus molecule is used, where it is converted to NADH plus H plus. So this is the second molecule of NAD plus. Okay, NADH. Again, succinyl-CoA is converted into, correct, no, fumarate. So here, correct, no, succinyl-CoA is converted into fumarate. That is, succinic acid is converted into fumarate. Here also dehydrogenation reaction will take place. Here it is not NADH or NAD, it is actually FAD. FAD will oxidize this reaction, where FAD will be converted into FADH2. That means 2 hydrogen is liberated. Understood? So again, the succinyl, once we have fumarate, fumarate is converted to what? Malate. So in the process of malate, correct? No? It is converted into malate. Again, malate, when it is again dehydrogenated into oxaloacetate, again one more molecule of NAD plus is used to convert into NADH. So during this process, again, we have one more hydrogen molecule. So here, whatever the hydrogen that is released out of this Krebs cycle is used for the electron transport chain. Very, very important. Understood? So this cycle continues. And then what will happen? We will be, this is called as almost half of your aerobic respiration is complete. Okay, by this. And then the hydrogen that is released will be used for electron transport chain. Okay, and this cycle will be happening in a cyclic form. That is why we call it as Krebs cycle. Whatever the molecule that enters here, okay, as acetyl-CoA, it will be eventually converted into oxaloacetate, and whatever the process of conversion of this is happening will again has to come back to oxaloacetate state only. Understood? That is why we call it as a cycle. So as the cycle keeps happening, the carbon chain will be reduced by removal of hydrogen atoms. As the hydrogen atoms are removed, the hydrogen atoms are used for electron transport chain, okay, with the help of oxygen and hydrogen. So the further steps of aerobic respiration can continue. Is it clear? Did you understand Krebs cycle? Students, did you understand this? Okay. So if you did not understand, you can just put me, in a chat box. Actually, we have to discuss this one more time in the revision class. I will discuss very clearly in the revision class. Okay, fine. The question here we have is the overall goal of glycolysis, Krebs cycle, and electron transport system is the formation of ATP in one large oxidation reaction, sugars, nucleic acid, ATP in small stepwise units. So you can take 1 minute to answer this question. You can type your answer in the chat box, okay? So the students from the YouTube, if you have understood or not, or if you have some answers to state, please try to do it, okay, in your chat box. Students, what is the answer for this? Try to answer. So the overall steps that are involved in this process, okay, what is it resulting in? Does it want to produce ATP in one large reaction at a time? Or it needs to produce sugars or nucleic acids or ATP in small step units? Very simple question, no? Give your answer in your chat box. Faster. All right. So I have a technical issue with battery, so there is no power. So we'll conclude this. Okay. The overall reaction of glycolysis is actually to produce the ATP molecules in as many small steps as possible. Why? Because at a time, if the glucose is broken down, tremendous amount of heat will be released and the capturing of energy would be difficult. So the slow reduction process of glucose will result in formation of, sequential formation of ATP molecules, so that the energy that is being released out of breaking the bonds can be conserved by forming the bonds or making the bonds in the form of ATP molecules. Okay. Sorry, students, I have to conclude the session a little bit early. Okay, 10 minutes early because of technical issues. There is no power. Okay. So we'll continue tomorrow in the next session and we will discuss the rest of the unit that is left out. All right. So we can end the session now. Thank you very much for this class. See you tomorrow.